This video is about macroevolution. And what we're going to do is use this acronym, FAME, to see some of the evidences that are used to support macroevolution. Do keep in mind that scientists look at the same evidence, but often their interpretations are very different. And I'll give you a few examples of that as we go through. But let's just take a look at some of the major evidences, and we'll evaluate those evidences for macroevolution. To give you an idea of macroevolution in a nutshell, we're going to start with this artist's portrayal of the early Earth. So the idea is that in the early Earth, there's lots of ammonia gases and sulfur and volcanic eruptions and things like that, but there's no life. And the planet is a hot, hostile mess. But it's within these conditions that the first amino acids begin to form. So based on this assumption of this is what the early Earth is like, there were some scientists who designed an experiment to see could they form amino acids. And again, the assumption is if amino acids would form, then proteins would form. If proteins form, then that's a molecule of life, and it could indicate how life started. So the experiment modeled the early Earth, and they were successful. There were some amino acids that, in fact, were formed. But to go from protein molecules to life is still quite a long ways. It's a huge, there's a huge gap between that. More interesting is that for amino acids to form, it's one thing, but for a protein to form, there's the necessity of an information-bearing molecule that tells that protein how to form, that tells those amino acids how to assemble. That's DNA and RNA, information-bearing molecules. So this experiment just simply pointed out that amino acids could form randomly in the right environment. That was very exciting because it supports this idea that simple things can lead to more complex things given enough time, chance, and natural processes. So if the proteins can assemble into something living, then we start with the first life forms that gradually branch off into other more varied and complex life forms, depending on the environment. So it starts very simple down here at the bottom, and it continues to branch as things are different and as the environmental conditions select for different things, so that we have all kinds of things. This is often referred to as an evolutionary tree. All right, so it starts very simple and it moves to things that are very complex. Again, it's important to notice that the tree isn't linear, but it is branching. So there's, we would say in this model that fish and echinoderms have a common ancestor way back here at the beginning of time. All right, so we wouldn't say that fish came from echinoderms, but we would say that they have a common ancestor. So again, there's evidence that supports this model, but different scientists look at that evidence differently, and different scientists look at that idea of how life began differently. So for example, fossil evidence, we would see that more simple things are at the bottom of the fossil layers, more complex things at the top. We would say that simple things, less complex life forms, are lower, so they're older. So the assumption is that the bottom of the rock pile is older than the top of the rock pile. Interestingly, there are other explanations. So one thing to consider is, how do fossils form? Fossils form not because something died in your backyard, not even because you buried something in your backyard, but the thing that dies has to be buried rapidly, and there has to be a lot of pressure. To explain that, it makes more sense for things like floods, landslides, volcanoes, and natural disasters, creating fossils. When those fossils occur, again, we do have simple things on the bottom, more complex things on the top. But one explanation would be that it's more mobile things at the top. So whenever there's a mudslide, whenever there's a volcano, as all these things are running away from that disaster, the immobile things are buried first, and the more mobile things, as they're climbing up or as they're running away, are buried last. Something else we find in the fossil record is not that there's simple things on the bottom and we get more branching and an increase in diversity as we go through time. Rather, what we find is in the Cambrian rock layers an explosion of all kinds of different life. And then, over time, we see an awful lot of extinction. So rather than the emergence of new species, the fossil record gives us an emergence of a lot of things all at once during the Cambrian explosion, and then this pattern of extinction through time, so that we see less things now than we would have 1,000 years ago. So this kind of illustrates the one model of looking at things where you have this common ancestor branching through time to lead to more diversity. But often what we see instead is rather this branching in the fossil record. We see a species, and then again higher in the rock level, we see the same thing. So rather than branching, that species continues through time. Next, we'll look at anatomical structures. We'll look at these three things: homology, analogy, and vestigial organs. So homologous structures, analogous structures, vestigial organs. Homologous structures just means the structures are the same. That is, they have the same structure. So if we take a look at all of these different living things, they all share a similar bone pattern in their forearm. So you have this like humerus bone, radius bone, ulna in the lower arm. And in a horse, you have something similar. And a cat, you have something similar, humerus here, radius ulna. In the porpoise, something similar. And the bat, something similar. So they're homologous. In other words, there's a similarity in structure. Analogous structures are similar in function. So we have like a moth wing, bat wing, a bird wing. They all help flight. All right, so the structure is not the same, but the function is the same. They all basically do the same thing. And again, different scientists interpret these differently. Some would say, well, that points to having a common ancestor. Others would say that it's that repeating pattern does not necessarily mean there's a common ancestor. It's just a functional design. Sometimes hands of primates are brought up as evidence of homology or similarity in structure. But a lot of times it depends on whether you focus on the similarities or if you focus on the differences. If you focus on the similarities, you would say all these hands are very similar. They all have 5 phalanges, and they all have similar bone structure. But if you look at the function of the hands, you'll see that there are significant differences. For example, the gibbon, the hand, functions very well for swinging, whereas the human hand is better for dexterity with that movement of fingers and thumb. So just because there's similarity in structure does not mean that they have the same functionality. The idea of vestigial structures is that this used to be some kind of useful organ in the past, but now as the organism is changing, this structure is no longer necessary. For example, hip bones in whales. The interesting thing about vestigial structures is that some things that we once considered vestigial, we recognize that they do have a current functionality. For example, the appendix in humans does have an important function for immunity. So when we look at something vestigial, it might be premature to say that it serves no other purpose, or it might be premature to say that it used to be something else, or even in this case, to call it a hip bone as opposed to something else, because it might have some other function we don't yet know about. So F for fossils, A for anatomy, M for molecules. For molecules, we'll mostly look at similarities and differences in DNA sequences. So if we read this chart going down, that means the human shares about 79% of this one small section of its DNA. All right, so this DNA codes for a gene, and we're just looking at that one gene to see how similar it is in other living things. So humans and pandas, about 80% similarity in that segment of DNA. Humans and turtles, 75, sea urchin 60, beetle 59, and yeast 50. So if we read it this way, we would say humans are more closely related to pandas than they are to yeast, because there's more similarities between humans and pandas in their DNA than there is between yeast and humans in their DNA. However, if you read the chart across, it tells a slightly different story. And that story is that sea urchins are just as different as they are from humans as they are from pandas. Like, we would expect sea urchins to be more closely related to pandas than we do to humans, but we find there's about the same, 60, 62, 63. And when we get down to yeast, we find that those differences are equal between all things. So rather than humans and yeast being very different, but humans and beetles and yeast being very similar, we find that yeast and beetles are just as different as humans and yeast. So rather than a branching picture, it looks more like a picture that's they're equally spaced apart, equally different apart. So that's molecular evidence. Now let's look at embryologic evidence. So the idea is, at least when it was first proposed, the idea is that embryologic development kind of mirrors evolutionary development. That is, starts simple, gets more complex. And the similarities between the pig and chicken at early stages represents their common ancestor, but then gradually they branch off and become different. The problem is that the more we learn about embryology, the less this holds up. In fact, there's vast differences even at these very initial levels. So for example, whenever those cells are forming, that blob of cells that you see, letter B, that's actually several layers of cells. There's an outer layer, middle layer, inner layer. And when you look at a chicken and a pig, those layers do not become the same things. Like, for example, if the outer layer in the pig became the brain, the outer layer in the chicken would not be brain tissue, it might be muscle tissue instead. The layers kind of matter because they become different structures. What we're finding the more we learn about embryology is that those things aren't as similar as they might look if you just saw them drawn or sketched in a diagram. So those are some things to consider about macroevolution. Again, there's a whole body of evidence, but different scientists look at it differently. And based on those differences, they start to form a model of how they view how life began and how it's progressing.